species. Yeah, let's go ahead and continue. I think they gave us new waypoints to head. Oh, we need a Niskin bottle. I was just thinking about it a minute ago.
Yeah, and then there is another third colony of the yellow.
Where?
A third colony of the yellow one. So just grab a piece.
I'm not.
For Niskin now?
Yeah, go ahead and get the Niskin now. What is it you're talking about?
There is Anita, her third colony of this, her third piece. Can you collect it?
What piece?
You need, she needs 3 pieces of coral.
Of what coral?
I think she wants these ones.
There's 4.
Okay. So that was a skin bottle 4? Thank you. Alright, sorry, I'm trying to see someone wants something.
Wait.
1837 and their depth is 1036 meters. I think we can.
We're good?
Yeah, let's just move over a little bit and get, I think we're going to collect another colony here in a little bit, but let's go ahead and move along the track because we saw more. 4. Yeah. I think we're still lateraling over kind of around 6, and then I think I'm not sure if we go from.
So I can face this big cliff and just kind of follow it along?
Yeah, let's just follow it along. I know. Yeah. And we got to the end of this last. Are the lasers back on? Alright, perfect, thank you. Yeah, I think when we got to the end of this one, we saw another one, so we can follow that one too and see. We can follow the trail of sponges.
Is that above me?
Yeah.
Wanna go up there?
Yeah, let's follow
There's
the.
like an overhang. I'm gonna be under it. Look how deep it goes too.
Wow.
Creepy. So creepy.
Let's follow the trail for a little bit before we keep going to the left. There's something. Is that a Drosemia that's. Yeah, it's a Drosemia. Yeah, then we can probably follow this back to the left now. Yeah, so it looks like we're reaching the top. Yeah. That's why sometimes I like when we just see what we see and then follow what we see. It makes it kind of fun.
Sometimes it makes sense, you know?
Yeah.
Only so much of the waypoint.
Yeah, and you can be just at the waypoint. You can be just off of where the most interesting part of a feature is.
Looks like
So-
here, like we lived the landscape sideways or something. You know?
Yeah, it does. You can see more abundance of the corals are coming in here. It's actually what. I'm gonna get a zoom in on that because that looks. I can't tell if it's a Drosemia here or if it's a different one we've collected. I think it might be a couple colonies of Drosemia, just white colonies. Yeah. And then we're seeing that other form of the Acanthogorgia where it's primarily one main axis and not as much branching. We collected one earlier on the dive. Oh yeah, look at that. It's beautiful. All the white Drosemia there with the sponges. I know, but we're gonna get it later because I don't want them all from the same place.
Okay.
It doesn't do me any good.
Wow.
Yeah,
Look at.
That's cool.
this is cool. This is probably another one where we should take the lasers off for a few nice shots with the big sponges and corals
That's cool. I don't care.
for Alex.
Hey, Zach, you ever seen a situation like this in a bunch of different files?
Not when I've been looking at files.
So you open up
What did you do? What did you do?
Upper Sparks, your shell goes to walls,
What are you trying to do?
looking like one side.
And if you go to USB disk, what happens? Click it.
No, but
Can't click it.
Well, you're in it right now. You're clicking on it.
So maybe
When you close it, you don't.
dealing with loading in the new waypoints, so sorry, just give them a moment. But yeah, to me this almost looks like a little kid scribbled with a crayon all over, and then someone did art right on top of it, like
Let's put some art on it.
yeah, putting in the other corals. Yeah.
Pretty zoom with this because I like this better than what Tyler did.
Yeah.
pay attention to that.
All right, let's go ahead and take the lasers off for some glamour shots. For these would make lovely wallpapers. For yeah. It's nice. You can see like different stages of the Drosemia being inflated and contracted.
Pretty cool.
Do this, but we can't access it.
Pretty cool.
So yeah, these are Drosemia. So Gersemia is the genus that I think they are. It's one of the what's commonly referred to as soft corals. It's not really valid so much because they're in different groups, like how they're evolutionary related, but Yeah, it's one of the genera that's known to occur in polar regions. Yeah. I'm gonna turn off the lasers and kind of pass through that again for.
Oh, shit. I thought they were off.
Yep.
Are y'all winning over there? No?
They're losing the battle to put in our new waypoints. So I'm just gonna do a nice slow scan here. Hopefully get a nice video highlight that Alex can use for some of the videos that you see in the expedition. Some good screen grabs. So this is much more similar to what we were seeing on yesterday's dive. So there's a tunicate up there.
Give it a shot. Where you look graffiti. Huh.
Would it be possible to like fly at that similar angle to the void, but then go up or go down, just so you get that like depth of field about just like how much stuff is on it.
Yeah. You can probably, actually, this one might be cool because we just came back last year with cool stuff.
Because I think we're just a hair exposed back just need to get into position.
How about something like that and flying up and down?
Yeah. And ideally have some clothes on the front, just like that, yeah. Would it
Wow.
be possible to fire us down this? Yeah, perfect. Just a hair overexposed on those fringes. Perfect. Thank you.
Yeah, those sponges are not good for exposure.
No.
Like
Well,
white
you like-
A lot
light.
of white things aren't.
How would you like bright light?
And then maybe another pass, assuming we just go up.
Okay. Facing the same way and everything?
Yeah.
We're gonna do one more pass for videography.
You've been standing there. Oh, this is an overhang. Crazy, I tell you, crazy.
Amazing.
Awesome, thanks. We can go ahead and put the lasers back on and continue on our way. Can you see what that is? It's a big red blob.
It is exposed stone.
Yeah,
sometimes.
exposed. Something came in. Yeah, let's do a zoom in on that. I think the geologist would love it.
Roger, park and zoom. Zoom, park and zoom.
Is it, or is that a sponge?
No, sure, no. What you see in a minute?
I think it's a sponge on top of the rock. See if we can see another one.
Roger, moving along.
Got some sponges.
Sangre llorando del documental.
Es mi especialidad.
Joel just watched the documentary at the last showing
Yeah.
and noticed that he said that he just saw Maritza crying, and she said it's her specialty. Yeah, Claudia had made Maritza
Nice
cry
workout.
every single time they did an interview.
The sucks.
So yeah, just as a reminder, until Tuesday night at 7 p.m. Central Time in the U.S., there is a documentary called Te Moana Ora that's available on YouTube. That is a documentary about one of the expeditions from last year, the one to Rapa Nui, with the kind of the perspective and viewpoint and importance relative to one of the Rapa Nui participants, Serafina Moulton. So yeah, it's largely in Spanish, but there's a version with English subtitles. And there's a version with Spanish subtitles as well for when people are speaking in Rapa Nui or English.
Did you get that all sorted out over there, Tyler?
No.
Not even
What's that?
not even a little bit.
I know a little bit. So right now it's with these waypoints, and we might know what they do.
Okay.
For whatever reason, we can't actually click on anything. That's not.
Man, there's just so much of this stuff. Look at all of it. That's crazy. What the heck? It's beautiful.
Yeah, it really is. That's impressive.
Where?
Are we landing on it yet? Not at the moment now.
Yeah.
Oh, yeah.
Oh, yeah. Do we? Yeah, we did the lasers back on. Perfect.
Yeah, this is like the roof.
Yeah.
I'm under this.
Okay. So we're actually like on a rock wall that's overhanging right now. So
Yeah.
I'm just gonna carefully navigate. Yeah, I'm seeing the little buffering signal coming up. So sorry, we've had some connectivity issues, I think, today.
Wow.
And we are working. Here's a yellow coral that we can take a look at and maybe sample.
Look at that.
Yeah, we've had some connectivity issues. We actually, before leaving on, really just until a month or so before coming on the expedition, they weren't sure how much connectivity we would have down off the coast of Antarctica. So,
Same with this yellow one, right?
yeah, we're gonna take a colony. Or a piece of the colony.
Did you want to know a fun fact about the internet?
Yes, I do want to know, and I'm sure the people at home want to know even more than me.
Well, we actually don't have a VSAT system like functioning at the moment. Obviously we have a VSAT system, but we're just a little bit out of range for the satellite that we normally use. So we've been only using Starlink, and the reason the stream was kind of bad this morning was because one of our Starlink antennas went down. So imagine all of the ship's traffic just going through Starlink for that one Starlink antenna.
Yeah, and you have to consider like the scientists on board. They're still
Mhm.
participating in meetings remotely, and
Yeah.
still we're looking up facts online as we're collecting specimens and everything else. Still trying to be productive when we're not on shift, and so that just adds more more traffic to the outgoing streams, incoming.
Yeah, and so that I actually had to, I haven't done this in a while, but I had to bump down the stream to 720p this morning.
Oh, wow.
Just so I could get anything out. So happy to be back in 1080 though. So, woo, woo.
Nice. Yeah, I think you're pretty much going to end up taking the whole thing or the majority of it because it's a pretty small colony. This one looks a little bit different than what we were collecting earlier, maybe similar to that small colony that I was looking at. It's sometimes hard to tell when they're small colonies. Kind of check the branching pattern just a little different. Yeah, isn't there something, Soph, about like they kind of needed to know where we were going to be to improve our chance of having good satellite service? She's busy. Nice. So yeah, it's about time for science party shift change. So this is going in this coral quiver. Perfect. This will be a nice specimen for sure for the museum too. With the holdfast intact. The waypoint entry going
It's on pause right now.
on pause for the collection? Yeah.
That should be good enough for a lid.
Yeah, I think you're alright to close.
Outside problems there too.
Look for the two for there.
Both at the same time.
I mean, this is kind of the like side scratch maneuver, right? Trying to like store the coral in your rib cage.
Not quite. Not quite enough. Maybe if I did this.
Alright, so there's a scallop in here, so I think it's
So
like
keep it closed.
largely keep it closed unless yeah. This is apparently actually empty.
Just watch out, you can go under
And
it.
so we still have 6 coral covers left. We'll let you know if there's anything else. We're getting kind of the last one that I think it's a different species, so it's good. I think we found a good one to collect for Antarctica. And there's a rock. There's apparently rock basket too. And that's one. I had to confirm today. I wasn't sure which one was which. But I think that's really it. They've done all the push cores. So
Suction sampler full.
suction sampler, no one told me exactly, but it looks maybe one. We'll have to confirm. And I don't know if there's anything they're willing to double up on, so you might have to check with them. And then we have 2 Niskin bottles that should be good till until 30 meters from ascending, so there's just one left. Yeah, and we've already gotten 1 year kind of like just sent me a wall and all that, so we're good. They're updating the waypoints. There's like a whole new plan, but they're having a hard time getting them put into the system. So they might have to manually enter them, but I think they're trying to do it again now before leaving. But essentially we're kind of like around that feature. We're kind of lateraling around it, and then I think the waypoints are gonna take us. Where's that map she sent? I think she's gonna take it to. Sorry, I gotta find the right chat. I don't know where she put the picture, but my understanding was like they're coming around. Like we're not going that direction anywhere. We're coming.
Okay, coming towards the camera.
I think so, but I haven't seen the final waypoints, and the map was a little confusing. Yeah, so they have to tackle that. So until then, we're kind of laddering left and following interesting features, doing zooms in. We've been doing a lot of videography for Alex, but I was told just to keep collecting and doing stuff like that for now until they had waypoints. So once we have waypoints, then we flip more into like following this stuff, and then they expect between waypoint 17 and 18 it's going to be limited visibility because we're going to be going downhill. So I think, like I said, I think that's when we're coming. Oh, nice. Oh yeah, pretty zoom there. So good control. Yeah, so that's my understanding, and then we've gotten a rock from the area that we were in, so I think everything else is followed from this page, and I'm pretty sure that these points just don't exist, and we're going to go from either 6 or 7 to 17 once they enter, I think, is my understanding. So, yep.
Pre there on goals, most people got what they wanted.
Yeah, I think so. We'll keep.
You've got the corals.
Yeah, we'll keep pointing out corals of interest. This is definitely the most diverse thing we've seen. We've gotten a black coral finally. So, Maritza or I will shoot a message if we want more as we go. Yeah, but I think we got at least Teresa's corals. Yeah. So yeah, that's where we're at. Yeah, great. Fun.
Oh, Tom.
The original
This stick work before?
No, the other stick worked and then it didn't work, and then I just brought this one in. Okay, and I can go get the other ones after.
Yeah, let's go for it.
Yeah.
Yeah, we tried that too. Nice. Okay, get rid of that.
You start this from the desktop. Desktop, you just can't do anything.
Get in there.
Get in there. Nice.
Hello, everyone joining the... Oh, everyone's already in the drive stream. I'm joining you. I'm new. We've just had a shift change. Maintaining this level of focus is quite exhausting, actually, so we're working 2-hour shifts. But don't worry, there's a WhatsApp group with everyone yelling their shopping lists at you. so that'll help out with the decision making. I'm Tom Linley. I'm a fish person from the Museum of New Zealand to Papua Tonga. If my voice seems familiar, I'm also the guy from the Deep Sea Podcast. And with me in the room,
I'm Pedro Martinez. Hello, I'm a Spanish marine biologist working in Germany at the Senckenberg Institute in northern Germany, in Wilhelmshaven. And yes, the shrimps, they have eggs. They are carrying the eggs in between the legs, and they are brooding them. This is an adaptation to make sure that the offspring has more possibilities to survive. This wall is amazing. How much life is there, the density of life, but also the diversity, how many different species we can see, different animal groups. Now in focus we see an ascidian, we see corals of different types. We see also sponges, little sponges, and also some brachiopods.
there's an amphipod living in that sea squirt.
Yeah, it is inside.
Nice little windowed home.
Yeah, sometimes they look for shelter. And there are also some worms, like polychaetes.
Oh, is that the one living with the coral there?
Yeah, exactly. So there is a tube. There's a white tube, a little bit to the left of the middle, and you see that there are some tentacles coming out of the tube, and there is also like a funnel. So this funnel is actually the closing mechanism. When the worm goes inside the tube, it will close with this little funnel that is there. These are serpulids. Serpulida is the family of the worms that we are looking at.
I didn't know that. That's great. It's like a plug on a chain.
And on top of the coral, we see also another worm, a polychaete. It looks to me like well, I am not a worm specialist actually. I don't want to say something completely wrong. So we are now at 1,028 meters. The dive started in a small sediment plain, also a little bit more than 1,000 meters. Then we moved some hundreds of meters and we encountered these walls, massive rocks, massive outcrops, completely vertical, and they are completely colonized by life.
Okay.
And this is because this is the reason why we are spending a lot of time here. Actually, our plan was to be by now already at 500 or 600 meters. Here we see also some bryozoans, these little white branches. These are bryozoans. And also beautiful shrimps.
Oh, we got two.
This one has some diffracting light in one of these antennae, and it seems that it is shining blue. This is probably not bioluminescence. This is probably the reflection of the little bristles that are in the antenna. But we cannot tell for sure. Maybe that is also bioluminescence. It's not my impression here. I think it's the light refraction.
It's these little guys, but there's a lot of them. A lot of them on this wall.
Here we see again a very nice serpulid worm, a little bit to the right in the view. This tube where we see some tentacles coming out and a little funnel and a little structure. And as I mentioned before, this little structure is used to close the tube when the worm goes inside.
You want to find out which tube?
So any of these little.
There are many of these
bivalve
tubes,
shells.
of these circulated tubes.
Slope, yes.
The black color of the rocks is probably manganese oxide. After a while, all the rocks are covered by this blackish manganese oxide
Yeah, no problem. They're not very-
minerals.
Fine, plenty.
And the lower part of this rock is also completely covered by life. We see very big sponges. All these sponges belong to, these are glass sponges, they belong to this Exactinellida group. Very interesting. Sponges are filter feeders, but this is also the case for most of the organisms that we see here, the corals, but also these circulate worms. And we see that there is also a lot of marine snow in the water, so they get enough food to survive, to grow, to reproduce. Even if, again, we need to remember that this place was covered with ice only a couple of weeks, couple of months ago. And the ice have been there for thousands and thousands of years. It was not very clear to us what to expect when we planned the dives. I was expecting to find much less life because
2 and 3.
the idea was that this area was not receiving energy, was not receiving food from the water column. But it seems that there has been a lot of lateral transport.
Then you put this on 3.
We will try to collect some of the brachiopods. The brachiopods, they grow, and in the shell we can recognize some growing lines, so that by inspecting, by analyzing the chemistry of the shell, and this is also something that we can date,
Could that be a pain? Could we do two?
the different growing lines on the shell, we can reconstruct the parameters in the water, we can recognize, reconstruct how much oxygen and how much, what are the properties of the water in different times.
Which is a good angle for you. Okay, that's one right there. That one over there, big bubble.
There is a question in the chat asking regarding if there is a volcano here. We have not detected any hydrothermal activity.
3-4 inches away from
This would be very interesting for us to, if we can find such a place, but for the moment we have not detected any fluids, any hydrothermal activity. In general, yes, in Antarctica there are several volcanoes, but not at this place that we are now.
There you go.
Somebody was past
Yep.
it.
That went. Okay, reducing section. And what's the next one?
Zoom out a little bit. So there's one
There's
there.
one over there. Yes. Not all. There you go. 2. Hold on. Let me get a better picture. You're about 6 inches away from the wall.
It's funny watching them snap closed.
You what?
Oh, got one.
You're about 4 inches. That's suction starting now.
So these are brachiopods. They look like mussels, but they're a really weird little group actually. Little outliers from the mollusks. But actually, it's not a biologist that's interested in these. It is a geologist, because these are incredibly slow growing, and because they bind up the local minerals to make their shells, they contain a history of the area, of the water chemistry, of the water temperatures, and so these little guys are scribes who have been taking notes of the water for hundreds or even thousands of years, and that's very useful. There's a couple of big ones up there, or do you want to move position? Just taking the opportunity to grab a few.
I'm moving.
There's loads of them, so if there's a spot that's easier for you.
You wanna try it somewhere else?
Just that they're so dense here, I was going to get a
Let's
decent
move it.
replica.
Can you move that one a little
Some
bit?
shrimp coming out to keep an eye on us.
Brachiopods are sometimes considered to be like living fossils. What this means, living fossils, is mean that we know them also from the fossil records. Specifically, brachiopods are known from the Cambrian, more than 500 million years ago, and they seems to be regarding the morphology of the shell, some of them exactly like species that are extant, that are living still. Now we can find them. So therefore we say, okay, that's a living fossil. That's an organism that is today still existing, alive, but we know them from the fossil record. Of course, we cannot say for sure, we cannot know for sure if this was exactly the same species, because in the fossil record we just have the shell, and even if it looks very similar, we don't know how was the genetic material and how was the inner morphology of the animals.
Is that a cup coral in the lower center?
Yeah, exactly.
Did we want any more samples?
How many brachiopods do you have at the moment?
I was going to get just a couple more, and
Yeah.
then if we wanted the cup coral.
No,
No,
it
no.
seems to be dead. I don't know.
Oh, okay.
We had some of them beautiful where you. We have had some nemertini in the dives. At the moment, I don't see any on this picture.
It's the one on top, isn't it? Let's see the picture. Oh, you're gonna get that one?
Oh, I see this little reddish one.
Yeah, it's actually not.
It is quite round. Nemertinis, they are normally compressed, so they are flattened. This could be also some flatworms, but ironically, the most primitive flatworms are not flat, but they are roundish in body shape. Or it could also be some polychaete worm. Some of the groups have lose the keta. I'm not sure if we have collected some of them to look at.
This is a vertical wall of rock. There's even overhangs and little recesses. This is some challenging driving. This is, I think, our second dive on this recently exposed piece of seabed. Part of the ice shelf broke away very recently, and we're very lucky to be in the area and be able to sort of jump on that opportunity.
Go ahead, Rich.
Yeah, if you're happy, I'll change the heading 30 degrees to port.
No, that should be okay. Okay.
Okay, yeah, let me know if you want me to move at all.
With it.
ROV ops are a tricky operation anyway, because there's a vehicle at the bottom of the ocean, a long tether, and then a vehicle on the surface as well. So we always have to coordinate our relative positions. We have to let the vessel know if we're moving or stopping so that they can match our pace. But there is the added element out here of the vessel can be dodging icebergs and sea ice while we're totally distracted down here poking corals. So every now and then the vessel will let us know that they're going to have to perform a maneuver, and we'll dodge out the way, and then it's our job to follow along with them. Usually the ROV sort of sets the pace and leads the dive, but now there's hazards in the water that we're just going to have to work with. We've got an ice pilot on board, we've got plenty of people on the bridge keeping an eye on things, and we've also got a radar that's pretty good at picking up the icebergs as they move around. A couple of different types of ice bouncing around out there. There's sea ice, which is frozen on the surface of the water, and generally that seems to be wind driven and a little bit smaller. And then there's actual bergs that have broken off from the shelf, and they tend to move tidally.
That is
But that is a massive overgeneralization. They all move dynamically. It's funny, the two different types of ice can almost move against each other because they're responding to different forces. Because of course the icebergs have that huge amount that's under the water, so they're more current driven, and then the sea ice floats right on the top, so that tends to be more wind driven. And all of these moving parts, including a vehicle on the bottom of the ocean 1,000 meters below you, make this quite a technical challenge, but the crew have really risen to that. Is the laser on? Have I just lost it in all the activity?
All that off.
Can we pop them on and we'll do a bit of a transect? Oh, come. It's such a busy image, I can't see them.
Yeah.
Your eyes are better than mine, but so long as they're on.
See that? Show you. See that?
Ah, there we go. Right, I'm tuned in now.
Oh, yeah.
So we've got to conduct a little bit of a visual transect, and those two little green dots are our laser scale. They're 10 centimeters apart, and so that'll allow us to process this video later on and figure out the density and size of these animals. So those couple of green pixels are incredibly useful. It's hard to feel context. A lot of these animals, have indeterminate growth. They can be really big, they can be really small, and it's very easy to to lose track of the scale of things when you're down here. So that will mean that our data can be quantitative as well as qualitative, so we can actually work out amounts of biomass and see subtle changes in the density of animals. Oh, this is a bit of a cave.
Yes.
Just to let you know, Tom, a little bit overhang. Yeah. So I'm not too close to the
So give it, give it loads of room. Yeah, don't worry about it. If you want to get up and over, don't worry about it.
Yeah, we need to take care not to go into cavities, because of course the cable needs to go straight up to the surface. We can get into trouble if the cable is touching the rocks. We don't want this.
I think the stream watching gets the science camera, which is certainly the most exciting one. But we get a few different angles in here, and it's amazing how you can sort of lose orientation of yourself in space. Like when we were doing that slurp gun back there, it looked to me like it was right where we wanted it to be, and then I looked at another camera and it's actually sort of 10 centimeters away from what we're doing. So you have to look at multiple angles to try and figure out how to use these tools. And you zoomed right in, but this is a vehicle the size of a small bus, huge, very strong manipulator arms. So the finesse with which they're used is really impressive. Do you think this is a sediment slip? So it's where the sediment has rolled down this rock face?
I think that one of our duties is to still collect a rock.
Okay. Oh, well, that looks loose just ahead of us, doesn't it?
Do you want to try that one?
Yeah. Doesn't look attached. There was no shortage of rock yesterday, but the problem was they were all attached.
Do we have some space left in there?
We're pretty good with the bio boxes. That's a lovely rock sample. Fist-sized, nicely fit in the bio box. Obviously fallen down from above and settled there.
Maybe not.
Is that a tricky spot?
No, I can't move forward anymore. Let me try going down a little bit.
So we were chatting about before about sort of situational awareness. There is an overhang or something else that's causing us issue here. Looks like a nice easy rock, tempting, but there's things going on out of shot. This is a big vehicle.
That's my other side. Let's see what happens. Okay.
There's the retractable tool tray, or sometimes called the porch, that's out in front of the vehicle, so we can extend that forward. It contains all of our toys and tools and sample jars. but it also has these buffers on both sides of the rail. And for situations like this, it's good to extend it forward, and you can almost push against those to stabilize you against a vertical face like this. I can say all that, but I certainly couldn't do it. This is tricky and far more things than I could hold in my brain at once.
See if it moves first. Flash forward a little bit.
We're also going to constantly maintain awareness of the pivot points and the range of articulation on the manipulator arm. It's easy to sort of lock out or run out of range of motion on those.
Yeah.
See the one on top of the stick?
It's a little bit far.
No, it's so welded.
Maybe you can roll it down.
Does it move at all?
Oh.
There you go. That one moves. Hold on.
They both do.
Put it on. I'm gonna move up a little bit.
We've been tricked before when a rock looks little and then you move it and there's a huge amount under the sediment.
Yes.
We got a very big rock yesterday.
So that's Antarctic.
There's geologists posing with it like a fishing trophy.
So we know that moves. So we'll get that one.
We have not seen any garbage, litter, trash at the seafloor here in Antarctica. This is something that you can see in many oceans because of the shipping lines of the vessels. Here there are very few vessels coming down to Antarctica, and all of them are very aware that there is not allowed to throw anything into the water. Here we have the Antarctic Treaty, which is an international law agreed by the states that signed the treaty to not to exploit the Antarctica, but also to take care of the environment. This of course includes that you cannot dispose and you cannot put anything into the water, into the ocean. And also the different Antarctic bases from the different countries, they make a lot of effort to get all the litter, all the garbage out of the station and transport this by ship to their home countries.
It would be very disappointed if there was litter underneath this previously totally covered area. It would mean it had been swept in laterally. Although we know from all these animals that food has certainly been moving in sideways and getting in under this shelf. We are doing a microplastic study as well in some of these samples, so unfortunately things like that might be swept in the current. There doesn't seem to be many places untouched. The deep ocean trenches certainly seem to be full of plastic because it just funnels down there, it's got nowhere else to go.
To be honest, finding a bottle or a can or something or a piece of wood, this could be historic litter. This could be for historic expeditions, and in this case, we would also try to catch it and see where is this coming from. But in the place that we are now, I don't think that there has been any historic cruises here, historic expedition.
I think it was the Kermadec Trench. We found a really distinctive beer can, and we put it on the internet and got all the sleuths involved, and it turned out it was like this limited edition one, only from a particular place and time, so it allowed us to really pinpoint it. But it was, I think it was quite old. I think it was like a decade old.
Well, the Antarctic continent is surrounded by the Antarctic circum-Antarctic current, which is going all in circling all the Antarctic continent in clockwise direction, and is isolating these water masses from the other oceans.
Trying to
So there is very little transport of objects into the Antarctic water masses from the surface waters. The Antarctic continent is connected through deep waters. This is one place where deep water is built, is formed, because there is a very large Antarctic shelf, and here the water gets very cold. And when the water gets cold, it gets also very dense. So it sinks to the bottom and then goes through the continental slope of the Antarctic into the abyssal plains of other oceans. And this is what we call the deep water formation. This happens also in the Arctic, but it's very much more important in the Antarctic.
How about the button?
It's those deep currents that originate here that bring oxygen to the deep sea. So even though we talk about different habitats and different locations, the whole world is connected as this sort of giant engine. And so if we were to lose these currents, we'd lose the deep sea. That sounds a bit dramatic, but it's happened about 5 times in Earth's history that the deep sea has suffocated and had a total reset. It's actually a part of the reason we get things like oil deposits, because lots of things died all at once and there wasn't even enough oxygen for them to be broken down by bacteria. It got sealed away and then chemically changed over time, and that's where we get our sort of petrochemicals now. So there's been these catastrophic hard resets of the deep ocean at multiple times. I think we think there's currently been 5. You can certainly see them in the sediment record. You can see these mass die-offs, and there's been lineages and groups of animals that we've lost, and we've seen ones recolonize the deep sea after that fact. Certainly within the fishes, you've got sort of late arrivers and early pioneers. So there's the theory basically is that there's 2 major groups. So there's ones that have really had enough time to alter their bodies. So things like the deep sea anglerfish and a lot of the other deep sea pelagic predators. They were one of the first ones to come back into the deep sea, and that gave them enough time to come up with whole dedicated organs, and their body has really changed. And then there are sort of latecomers which look very much like their shallow water counterparts. So the snailfish would be a great example of that. A Hadal snailfish doesn't look that different from one you might find in the shallows, and it doesn't necessarily mean that they're not so good at pioneering the deep sea. Certainly in the case of the snailfish, they just seem to have something about them makes them very, very good at going deep. They're the world's deepest fish. So it doesn't matter that they came late, they still did quite successfully. But you see these two main groups. One has totally changed the body structures and even has new structures adapted to the deep sea, and the others have just made the required chemical changes basically to be able to cope with the high pressure, and so that's allowed them to pioneer their way in as well.
The deep sea waters in all oceans, they are cold. So even if you go to the tropics and start diving, you will see that in a couple of hundreds of meters the water gets cold, and if you go to the deep sea, 1,000, 2,000, 3,000, 5,000 meters, the water will be only a couple of degrees, and it will be fully oxygenated. And this is something that people ask us in conferences, why the water is so cold there and it has so much oxygen. And the reason is that the water is formed here in Antarctica. It's a deep water formation by cooling the water, and because it's surface waters, it is has a lot of oxygen. So this water circulation, the water will come up to the surface in upwelling regions, and the water circulation from its beginning in Antarctica here to coming up to the surface again will take some thousands of years. between 500 and 1,000, 1,500 years.
So the oxygen that deep sea critters are respiring can be hundreds or thousands of years old.
Yeah.
Which I think in the early days gave us some surprises when we did chemical analysis, because some of the dating methods showed them to be
Very
really,
old.
really old. We couldn't figure out why.
Yeah.
Huh?
Yeah.
I thought it was.
Let's try this. Yeah, it's actually, it's not a problem anymore. So all we can do for now is carry on, I think. Do they need that for something right now?
No, we were gonna pick up that one.
And you tried restarting it, right?
Yeah. Restart again.
Turn it off on the,
We are still trying to pick up that rock. It's a little bit difficult because it's a bit away and we cannot
No,
approach it.
I'm still talking to her. Your back looks like that.
Oh, yeah, this is what the one was, okay.
You got your own. Now I'm off. Seems like it's the, not one.
Switch.
So it seems like.
Oh, yeah.
Alright.
Just swap. So that's swap it.
Okay. So in the meanwhile, if you can get it with the one with the blade on, you can do that, but if not, then you just can't get it right now.
Try. Twice.
So once you put this up, it's not right.
Let's not use that blade.
Yeah, we're gonna.
I think we're gonna have to find a smaller one.
Yep.
I don't know. Push that one out.
Yes. So we are going to move on.
It's,
This rock is also by far too big.
yeah, I'm gonna move on.
No problem. rock bested us. We're having some issues with the right manipulator arm, so we need to find a rock that will fit in the left. Very unusual fish just went past. I only caught a glimpse of it. Yeah, just head towards the waypoint. Zoomed out, lasers on. We'll transect it. Challenging environment. Is that a bryozoan there? Lovely, lacy.
Yeah,
They're really pretty and delicate here.
very beautiful one. Okay, we continue. We want to surround this outcrop. In order to map, to do a visual census of the communities that are living here.
Each of these sponges seems to have a little resident shrimp. You can see something red in the bottom of each one.
Yeah.
No, don't do it right now. Because our umbilicals towards the tail end. In fact, move them east. That way. Yeah. Because now our umbilical is running close to the rocks.
It's easier, feel free to come up to the rock.
Also a big sponge
ROV break.
in front of us.
Yeah, we have rotated by half a unit hours. Just getting underway.
That's great. It's going to be some complex analysis to work out this area.
Yeah.
Nice sandy seabed, you just use the ROV speed and altitude, but this has some shapes. There's some fun new analysis. We've had photogrammetry for a long time, but you sort of get it's called structure from motion, so you can use frames of the video to actually create a bit of a 3D model of these spaces. And the technology's really come on. When I first got into photogrammetry, it was very, very finicky, and it was easy to make things explode out in crazy directions. But the algorithms are quite forgiving now. But still, this is complicated, and any variations in the camera optics and any changes in light source, it's really challenging. But it's very, very fun to take 2D images and make this 3D reconstruction. And then you can work out surface area, and then we can work out actually what volume of habitat this is with our handy laser scale. There seems to be this channel in the rock face, and obviously sediment has flowed down it, so sediment from higher up has just been flowing down this channel. You can see that it's rained down from above as well and collected in here. So there's probably a a sedimenty patch at the top here that's just continually trickling down onto these lower rocky plains. I think we're entering the sediment area now. Yeah, I think we're just coming up on the peak of this
yes.
rise. Really complicated environment. Not easy flying.
Yeah.
In the big picture. Zoom out.
Everything colonized. Very beautiful.
This is something. Keep going.
I think so, yeah, keep heading to the waypoint.
We see these cap corals now with the polyps out. I think these are the white corals with a single polyp. It's difficult to, I mean, I had never seen them alive.
Shout out if there's anything you want to grab.
There is a sea urchin. A lot of shrimps also. Rocks. Oh, an arrow worm.
Oh, a sea spider on that rock.
Oh yes.
You want to see it?
Yeah, zoom in on it just to get some nice shots.
I think that we already had a sea spider, is correct?
In this collection, I'm not.
Yeah, I did see a sea spider.
Had we collected it?
Yeah.
Oh, okay. I'm not seeing it.
I mean,
Nice. Sea spiders are usually quite small in all the other places in the world, but they get particularly large in Antarctica. So that one's probably 20 centimeters across if you extended all those legs. They get even bigger here. Creepy and spindly looking, but unless you're a sponge, they're nothing to worry about.
yeah. Oh, it's beautiful.
That's great.
I learned that in English they are also called
Yeah.
nobodies. Is
Yeah.
this right?
What's that?
Nobodies,
Nobody's.
because they don't have body.
I haven't heard it, but I like it.
I mean,
It's all leg.
It looks like the legs insert in the middle of each other.
Yeah, do you have
Okay, this is on all 6,
Okay.
all 7, 8, 9.
And how do they relate? Sorry?
Yeah, I don't know about that. I don't know about this.
Okay.
Sorry.
No, no problem. Is it? I know.
Yeah, that one. This is after 8.
Yeah.
Okay, so it's quite a different location, so I can
Yeah.
almost eject that from the
These are the
brain.
new ones. These are the new ones, right?
I think these are all.
Yeah. So she's graduated?
Yeah, I think the new points skip directly to 17. I just wanted to make sure I was doing the right objectives.
That's actually what is happening.
Okay, it's a bit more complex.
Yeah, I think nothing. I mean, there's no 17 or 33.
Yeah,
This one.
that's why I thought maybe the numbers jumped lower, and so 7 is 17.
I'm also a bit confused.
Now we got back to a more
Some
sedimentary
other
area
objectives,
where
that's
we
okay.
see this sea
I don't know what to be
urchins,
doing.
the little red roundish
Grab any
organisms.
No worries.
And this seems to be also a sea urchin, the white one, but might be also something different.
Right, Charlene, that's the new updated red one. Or small one.
Reach the apex of this particular ridge, covered in sediment on top, so all that sediment we saw cascading down the rocks, this is where it's coming from. And is it just that we're moving quicker, or does the current speed look a little bit higher to you? There is current, okay. So these seabed forms modify the current regime. It makes it the sheltered spaces and there's windier spaces, and that's really going to impact the animals. Some of them like plenty of flow. Their bodies are strong enough, they can hold onto the rock, and it brings the food to them. They don't have to work very hard. Some things are quite fragile and like to gather the food themselves. Also, some are quite picky. Some just have a great big filter that they hold into the current and they'll eat anything, and their gut sorts it out. And some are picky eaters who like to be able to select what they're eating, and having it blasted into your face makes that difficult. There's two main feeding guilds we'll see down here. There's sort of sediment feeding detritivores, so all these animal tracks you can see on the seabed, that's where things have been shoveling up the sediment, the food sources that settle on the bottom, all this marine snow. And then on rocky areas, those things that hold filters up into the water column, essentially the filter feeders. So that's the two. They're still both exploiting the marine snow, but the two foundations of this food web, they're the ones at the bottom, and then you'll get your predators and parasites and things that live up on that. See all these animal traces, see the mounds that worms have excavated, so we know there's a lot living in the sediment here, and all these little trails of grazers who have trundled along, like that sea cucumber, that sea pig in the bottom centre, very, very slowly trundling along, snuffling up all that biological material on the surface. So you can actually see the colour. The clean sand is a lot paler, a lot cleaner looking, and then the ones with this sort of golden, greeny coating, that's the good stuff. That's what they're after. You can see a little urchin down there as well, and they see exactly where it's been cleaning up.
sea pig.
Good chunky ones here. On our shifts, we've usually been hunting them, so it's hard to just let them go by when I see a nice juicy sea pig.
By the way, we also should collect the sackito of sediment.
Okay.
I think there is one. They have collected one. I think we could collect another one.
You want to do that here?
Yeah. Is this apex
Yes.
a good spot? Yeah. Let's go for it.
Spot.
And then we get it on the position.
What's this here? Is that a drop stone?
It might be a good size to collect.
Let's see.
I think we're at the apex, so it must have been ice transferred. But yeah.
See, that thing was controlled by
We've been tricked in the past with a tiny looking rock turning out to be huge, or even just attached to the bedrock, so a cautious poke usually reveals things. Whoa,
Oh yeah.
perfect. Once we have it, if we can hold it in front of the camera with the scale, give it a glamour shot. I'm still not over being outwitted by a rock earlier. I am coming back with a rock. This is not our preference of manipulator arm to pick up a rock. So we're a bit like a lobster. We've got two different claws, different tasks. This one has the soft manips, the yellow and the red you can see there. It's a little bit gentler. It's good for plucking coral. And if you look closely, there's actually even a cutting blade on there as well. So we certainly don't want to dull that on a rock. It's our pruning scissors for snipping little bits off a coral. Can we give the claw a spin? Can we give the, yeah, just get it in context.
Spin.
Nice. Oh, the
other side.
There seems to be a drop stone, no?
I don't see another way it could be up this high. I think it must be a drop stone. It's great. We've got a few bio boxes open. There's a scallop in BB4, so I'm nervous to open that one.
What we could also do is to put it in the sackito, the stone,
Is that okay? Do they not miss out on
together
sediment
with the
then?
sediment, because they will sieve it later on. If this helps, I don't know.
Yeah?
There might be a problem with the sites.
Yeah.
I just
Oh,
realized
because the arms
the
are.
arms are looking on the sites.
Right. Okay. So we're going by hand. And same with the sort of rock baskets, we won't have the range to reach over there.
We might be able to. Let's take a look.
We've got other options though. We've got maybe BB2A. I don't think there's anything in here that's going to escape.
See if you could, yeah. Do you want to put it in the rock box?
I think that's appropriate. Yeah, it's a rock.
Okay,
I've not used the rock box before.
I'm just gonna watch your shoulder, right? see the picture. You can't use the scuba, so well, clear. Still clear. Still clear. Still clear. Okay, not touching.
I don't know if I'm on top of the.
What's the yeah, that's the most max you're gonna get. Okay, hold on. Shoulder up again. Okay. That's the wrong box. Still good. Watch it. To go. Drop it off and watch it. You can drop it over there. Okay.
Okay, it's inside.
Okay. Nice job. Okay.
Right, so very good.
Well done. There is another rock in there, but they're quite visually distinct, so we'll be able to have them share a space. I don't think rocks fight. If we're the first to record rocks fighting, then we've got a nature paper. Maybe they'll breed.
So what other options do we have here?
The
You
second.
want a sack?
Yes,
You can reach.
if possible.
Swap. Try swap.
Let's swap with the sacks. Well,
Sacks are on the right.
levels.
Hold on. There might be a sack here.
You can also put the sea urchins also in the sack together with the sediment.
There's one.
I don't think the geologists like stinky biology getting in with them.
No, the sack is for.
Oh, is that biology?
Yeah.
Oh, right, it's you.
That's for medical.
Oh, right, okay, okay.
That's what I'm saying. I don't think you can reach that.
Does it have the right tool grip for the sacks?
The what?
Does the left arm have the right tool grip for the sacks?
I don't think he has to reach.
Oh, this here, right?
Bear with us for just a second. We're going to see if we can solve the issue with the right manipulator arm. I think it's vessel side, so it might be something we can fix from here. A lot of the rest of the dive is, well, we're after rocks, and we want to take some sediment samples with the sacks, and unfortunately that's on the right side, so we're missing that arm. We could adapt the dive probably, no problem, based on what we can reach, but if this is a simple fix, it would be great to have it back online.
We are right-handed. We prefer to use the right hand.
Well, it's the big chunky one. It's the big strong smash rock, grab sediment one. The other one is our pruning scissors for corals.
This Puskora 18 seems to be empty. I don't know what happened.
It's marked as oh no, 8 is marked as geo. It's labelled as if it was taken. Might just have quite a lot of water. Yeah.
Very nice.
We're back, baby. Yes, please.
Do you want to do some first?
Oh, the rocks are terrified now. Yes, please. Easier.
I was thinking on Darth Vader from Star Wars that also lose the right arm.
Ah, yeah. And then immediately gets a robot hand, and it's
Yes,
not really a plot
exactly.
device at all.
Okay. I'm gonna start with that.
The chat has named the manipulators Snippy and Grippy, and I love that, so I hope that sticks.
Did it work?
Yeah.
So it is the controller for sure?
Yeah.
Okay. Well, luckily we have another customer.
Oh, well, the switch is working too.
Look, we're asking how the conditions are up at the surface. It is an eerie calm, considering something quite major happened here recently, and the shelf broke away. It is glassy on the water surface, huge, beautiful, freshly carved icebergs dotting the landscape and giving us a little bit of a sense of location. Yesterday, at past midnight, was a beautiful sunny day. I am not getting used to this 24-hour sunlight. It's pretty nice because we're on night shift. It's pretty nice to not feel like you're a filthy night creature that no one ever sees, but it makes going to sleep difficult because you go outside for some fresh air and your eyes get blasted by sunlight, and that's it. Your body thinks it's morning again. So it was a beautiful sunny night last night, and then this fog just rolled in, and we delayed ops a little bit actually because we were worried about the visibility. Of course, we need to keep an eye out for all those icebergs, and we like to have redundancy, so the radar's pretty good, but we need eyes on them as well. So that fog rolled in, it had a lot of moisture in it, and the temperature just dropped. Really, really cold wind chill. I was out on deck bringing a piece of gear in and out that had some fiddly screws, so I couldn't wear gloves, and just the wind chill got you really, really quickly. The temperature's surprisingly, a couple of degrees below freezing, but it's like Antarctic cold is different. It's a real dry, loads of wind chill. So I lived in Scotland for 8 years, and I've felt some cold, but it feels different here.
By the way, Tom, how was your shift yesterday night?
How was my deployments?
Yeah, your deployment.
It was good. It was had a few tricky bits, had a few little technical problems, had a housing leak a little bit. It's annoying. One that I repaired worked fine, and then the other one started leaking, but it didn't kill the camera, luckily, because of the design. It's upside down, so luckily the water that did get in pooled on the other side and didn't wreck my camera. That was out for about 7 hours. This is a mid-water pelagic light lure, so trying to get some of the shyer creatures, and it's exciting, but it generates a lot of data. So I've got a 360 camera on this as well as a low light camera, so it generates about 21 hours of footage during a deployment, and at the end of it I'm pretty exhausted, so I've got to be honest, I back it up, I check that it worked, and then I try and get some sleep. I wrote a little script. I had a co-pilot's help. I wrote a script to scrub through all the video and just look for particular bits of activity. So that's saved me a lot of time. So I can leave that scrubbing for the video, which it's doing right now actually while I'm on shift. So it's looking through my video for me for anything interesting that comes into shot, and I get to see what I got when I get off shift in a little bit.
You can also grab this little sea urchin if you mind.
In the same set?
Yeah, sure.
Can you see him? Oh yeah.
He wants to see urchins.
Yeah, just put it in the same graph, in the same. Yeah.
He's gonna put it down, he's gonna be grabbing.
we can notice that we have some deep sea current. The very soft sediment is transported immediately away of our field of view.
Yeah, that's the picture.
It's moving to the right. So we are heading now 76 degrees.
While we have the sack out, we just decided that the sea urchin would like to come along. Oh, that's wily. We think this sediment might have built up over a very, very long period of time. It's kind of cohesive and cakey. I think it is a sand, but it's quite oozy. It flows, has a lot of cohesion. Sometimes when we're taking the push cores, it really clings to the outside. You can see how it's clinging to the sack. So the geologists will have fun backtracking all this history.
So we are heading about 77 degrees, and the current is coming from left to right, so probably it's coming directly from the north, heading south. We need to look into the map later on what this means exactly, where this is coming from, because this currents are also the source of food for these communities that were covered by ice for so many thousands of years. Well, if it is not possible, then we just leave it the urchin. The primary goal was to get some sediment.
Do you know where the sacks go on the sample list?
I don't know where
I'll
it
just
is.
write them out as a space.
Is that enough sediment? You won't get one?
Yeah, that's enough. That's good.
So we'll put this in the rock box. We'll put this in the middle, inside. Okay?
Okay.
It's a picture.
I think we've got a good picture of where it was taken. Definitely have it on the 10-second timestamp.
Yeah, 10 seconds.
I'm gonna let you see a picture of your arm, right? So that you can see the arm here. And what it means. You can see it.
Okay.
There you go.
They're fairly protected in there. They don't wash out on the way out. They're fairly protected in the rock basket. They don't wash out on the way out.
I don't know what's it. They have already collected one, but I don't know where they put it. But they are already.
Covering the handle. Should be alright. I think your satchel is covering the other sack. But maybe can you do
Okay.
rotate?
Yeah, that's the same spot.
Yeah, that's right. Let it go, yeah, that way. There you go. That should be good there. Yep. Bombs away. Okay.
the next task?
Yeah.
I think we transect on for a little
Yeah.
bit.
Transit.
Still, let me pull the
All good? Grab a few samples. Now we're going to have a transect of this sandy habitat on top of this rise, and eventually we'll start coming down the other side.
Yeah. I'm just gonna take them, just gonna test this.
We started this dive at about 1,200 meters deep, I think maybe a little bit more than that, and we've been climbing this rocky ridge, and we're up to about 1,000 meters deep now.
Yeah, he opens but doesn't close. Same as it doesn't close on Antarctic. It's kind of weird sometimes.
Hey, there you go.
Sometimes it is. Yeah, but sometimes like, because he's erupting, so now he's not.
Anyone just joining these dive streams, we have had an amazing opportunity out here. So just a few days ago, a large piece of the ice shelf broke away, and not just broke away, but very politely then got out of the way. So this dive and the previous dive, we've been exploring an area of seabed that has been covered by ice for thousands of years. There has been ocean under it, maybe 300 meters of seawater, so animals can get in and food can get in, but it's been cut off from the surface and sort of fed by these lateral currents. So we're really good opportunity to look at that pristine environment, to set a baseline for how that might now change now that the ice has gone from above it, and also compare it to the other sites we've already been that aren't under ice. And it feels like there's a tonal difference. It might not be that it's covered by ice, it might be that the dynamics and the hydrology are different because of the ice. Certainly seems to be plenty of flow and food coming this way.
Another sea spider, isn't it, Bodo?
I think we're okay.
Yeah, we're okay. Continue.
There's something fishy there.
What is that?
Fishy or wormy. I had a pointy thing, it's gone.
You think it's a fish?
I do. I think it's an eel pout. Yeah.
Go ahead.
And a sea urchin covered by sponge.
Sponge hat.
Beautiful, beautiful view.
Do you think we'd be able to slope the fish?
The fish?
Yeah.
Yeah, you can start the fish.
Fish might have something to say about it. We can try.
That one?
Yeah, but
Okay.
it might already be onto us.
It should be on still. Just keep your eye on it.
This is zoacid and eel pout. We've been seeing a few of them that we've been making fun of because they roll up like donuts when they're scared. But this looks different. I can tell because the gills are enclosed. The ones we were seeing earlier have the sort of traditional fishy gills with the operculum with that bony plate on the outside, whereas this one has enclosed gills. They're really abundant down in Antarctica. This is where this family are doing particularly well. They got down here from the northern hemisphere and then just decided, yeah, we're going to specialize in this, and then radiated into lots of different species.
The two dots in the center of the screen, the green
We're
ones
still on chamber 2.
is a scale. This is 10 centimeters apart. They're produced by laser pointers
Two's fine.
and help us to get a view of the size of the structures
yes,
and
yeah.
the organisms.
Okay, moving close slowly. Oh, sorry. Go for the front. You go for the top.
A vehicle the size of an SUV is now sneaking up on a fish about 9 centimeters long.
Let me move closer. Let me move closer. Okay, easy, easy. Just keep your eye on the subject, Tom.
Okay.
Bring your.
It's onto us. Shouldn't be a particularly strong swimmer.
Get in front of him. You can. There you go. See if you can get someone in front of him. Okay. Going slowly. And I'm going to turn this on the last moment. Keep coming closer.
Looking wary.
Keep coming closer. Keep coming closer.
Like I was choosing to walk rather than swim.
And then last moment, slip in. Coming closer. Coming closer.
Yeah, this is different than the ones we've seen before. And if it's specialized in living under ice, it might not be one we know about.
2, 3. Go. Go. Dip it in. Dip it in.
Outwitted.
Escaped.
Alright. Is he still there?
May have bolted away a fair way. Don't worry about it, I think he's got well clear.
Yeah.
Thanks for trying. He was onto us though.
We've caught plenty of fish before. This is Joel's first attempt catching fish.
They're advanced level. It's got eyes, it can move.
Oh, we've caught a lot of eels and things. It's just that you just need practice with it.
I don't envy it at all. I can just wave at a screen and make unreasonable demands. It's nice to give you folks at home a little bit of a visual. So where we're sitting, we've got banks and banks of monitors in front of us, but we've got these handy like extendable pointy sticks, and so basically we just point at the screen. I want that one. I want that one. But somebody obviously figured out that this is a lot simpler and causes a lot less confusion than trying to describe things on the screen, and we use technical terms that aren't very accessible, and nope, just a big pointy stick.
So we can continue our transect.
We got some nice shots of that eel part though. I'll see if I can ID it.
Forward.
They are a
Yeah,
really,
moving
really tricky
forward.
group. Yeah, continue to the waypoint. We'll go transect mode.
Transect mode.
We discovered 3 new eel pouts just off New Zealand last year as part of the ocean census expedition we did there. Oh, is that a crinoid? Yeah, that's really pretty.
Yes, crinoid. bioturbation on the sediment surface. A lot of traces.
They all tell a story. Some are wiggly, some are straight lines. Little deep ocean rumbas.
Now we see a very large brittle star.
It's already, that's no native weapon.
And again, lots of sea urchins. Anemone.
Seafloor sloping away to the left. I think we're just rounding the apex of this particular point, and then we'll come down the other side.
And the crinoids.
I'm used to crinoids sort of holding the arms up a little bit more into the water column, but these seem to be lying flat. They're waiting for things to fall on them rather than filtering, it feels. Maybe the current's too high here to put your arms up into the water. You have to stay low to the boundary layer.
Here another one.
That's quite steep. Another crinoid.
Oh, what's coming up? Oh, that's a sea star actually.
Continue.
Freyella, yes, continue.
That's an interesting pose.
They are also filtering,
It's current.
and that's a brittle star. We have a slope here.
A few more drop stones.
Oh yeah.
Rocks. You want to pick a rock?
No, I think they're pretty big.
They're very, if we find a smaller one, but we could try.
Now we have a working minute now.
Excellent. So since we're at the top of this rise, there's probably no other way these rocks could have gotten here other than being delivered by the ice. So they're iceberg drop stones. So as the ice shelf forms on land by loads of snow compacting down into ice, it causes a huge amount of erosion and it picks up rocks along the way. Then once they get out over the ocean, they might start to thaw and that rock wiggles its way loose, and it's the only reason I can think of that we'd have a rock sitting on the surface of this sediment. There's nowhere else for it to fall from. So they will be from the Antarctic continent. They will contain lots of interesting information. If we can find one claw-sized, we'll pick that up for the geologists. For similar reasons, Antarctica, well, the polar regions in general, are really good places to look for asteroids. So anything sitting on top of the ice and snow, a rock sitting on top of the ice and snow, there's no real other way it could have gotten there other than falling from space. So it's a really good way of finding asteroids and having some confidence
Meteorites,
that's where it comes from. Oh, sorry.
Meteorites.
Meteorites, yeah, sorry.
Yeah, indeed. We had in the last cruise that we had in the Atlantic, we were collecting the deep sea, and some colleagues from us, from another Senckenberg Institute, they are looking for meteorites, and some of the meteorites are magnetic, so they were using a magnet on the deep sea sediments in order to collect these micrometeorites.
Yeah. It's funny from an angle, isn't it? Oh, some nice traces here. Looks like the Nazca lines.
Yeah, it's from these acorn worms.
Directly south or east.
Maybe we try to pick up this one.
You want to try?
Yeah.
Yeah. Try this one.
There's a small sponge on it. There is also a worm, maybe an ascidian.
Place that's an updated with.
Yeah, if we can.
See if it moves.
It doesn't look attached.
It's kind
Oh, it's bigger than I thought.
of, yeah. Must be.
It's too big, right?
Yep.
Okay.
Okay. Moving forward.
Ah, it's too big for a stomach.
But it's maybe a smaller one.
What is that?
Oh yeah, is there another one there?
That's a smaller one.
There's a lot of drop stones. For all the help it does. There's a little isopod next to it.
Yeah,
One of those
maybe,
spindly ones.
Yes. Maybe this could work.
Okay.
It's good.
Okay.
Yeah, it's good to take a picture of the rock so that we can separate them later.
Yeah. And we've got to pick where to put it, where it's going to be clear which one's which. We've got a lot of space in Biobox 4, but there's a scallop in there, and they've been doing a really good job of escaping when we open the lid.
Yes,
So
yes.
I'll only use that if I have to. Or I'll drop a rock on it, that'll done it.
I think this can go to the other rocks now. So this isopod looks like a monopside. This family, they are good swimmers. They can go into the water and they have paddle limbs, legs that they can use for swimming.
Can we put that on the side box?
Yes, actually, we probably can.
Yeah.
Is there a glamour shot?
That's going to give it a flip. Yeah.
Yeah, it's a bryozoan on top of the rock here. Yellowish one.
Nice.
I think it will be straightforward to identify them, to separate the rocks.
Sounds good.
Yeah.
Okay, so we just throw in the rock box on the side?
Yeah, which one does it relate to? That one? Yep. There's a couple of other rocks in there.
Okay, you want to put in the middle? This
Yeah,
one?
yeah, that
Let's
one
put it in
seems to be
between.
clear.
And that. Okay, approaching up.
Rock basket too.
Everything's section.
Excellent.
Very good.
So that's in rock basket too.
Is in the basket.
Awesome. Oh, we're getting the rocks today. back into transect mode and we'll head to the next waypoint.
First yesterday.
Okay.
Unless I say something, but this is the research
Right, 2 of them are reserved now.
in the ascending. 50.
Nice view from outside.
Oh yeah. It's just glassy calm out there. It's beautiful. I see why people fall in love with this place. Like Antarctica is special. Done a lot of traveling, and this is something else here. Onward and upwards, transect mode, so we're heading to the next waypoint. Just had our pilot shift change. It's pretty exhausting maintaining this level of focus.
No, more rocks.
More big drop stones.
Big ones.
Go that way.
All the way down. Instead of moving all the way down there, make sure it's like all the way up to the next one. Like you give them a move, just let it be. You know what I mean?
Okay, Rich. Annotated weapon.
Updated quickly.
There's plenty of sea urchins at the bottom.
Yep, no problem. Yeah. This is pretty consistent.
Sea pigs. Or sea cucumbers.
Do you know what type these urchins are? We see a lot of them.
It seems to be irregular sea urchins. So it's sea urchins, there are 2 types. Ones that they are quite symmetric, so spheric, like a sphere, and others that they are more pointed or bilateral, symmetric. These are the ones that also go into the sediment and live in the sediment. But this one had the shape more like a bottle and is living on the sediment. we guess that. Let me check if I find it here in the book. In other places, there is one that is called Porturesia.
Sorry, stream. I was trying to play a DVD. The echinoderms have quite different body plans. Quite different body plans within the echinoderms. So that's the sea stars, the urchins, and the sea cucumbers. But if you look really closely at their structure, you can see how they got there. So if you took a sea star and folded all its legs back, all its arms back, until it made a sphere, you'd have a sea urchin. And if you then stretched that sphere out into a tube, you'd have a sea cucumber. And you can still see some of that radial symmetry in the other forms.
So in the Antarctic macrofauna atlas, we found the sea urchin that looks quite similar. This Portalesia hispida might be that species. It's has an oval form. It has this purple color. It could be that one.
Oh yeah, there we go.
Sea stars are a bit of a democracy as well. They've just got very simple nerve net and sort of ring of nerves around the central disc, and you can cut the arms off, and as long as you get a bit of the central disc, they'll regrow into a whole new animal. Oh, we're just entering the edge of this drop. So when they're responding to the smells of food or danger, the arms actually sort of make their own decision, and then the whole animal moves as a result of all those different decisions. But there is one lead arm that will make a decision if there's it sort of breaks a stalemate. And most of them have a little dark point at the end of each arm, and that's a sort of light collecting organ. And the arm that's in charge, it's usually a little bit bigger on that arm. So they vote democratically, but there is a lead arm to be a tiebreaker. And if that arm's lost, then when it regrows, one of the other arms might take up the mantle of boss arm.
Now we found rocks again. Outcrop.
So that was the apex, the sandy area, and now we're coming down the other side of this ridge. Really tricky terrain to navigate.
Some sponges.
We get sucked in and sort of watch from just the main science camera, but it's worth realizing that there's a vehicle the size of a small bus with a tail coming out the back of it that's incredibly fragile, and a tangle point connected to, 1,000 meters above a vessel that's dodging icebergs. So this is a multi-layered activity, and I just get to watch the camera and not have to worry about that so much. Oh, wow.
Oh, that's like a small coral or a sponge.
Can we have a zoom in if it's an easy spot to maneuver? No, we need to get that clear. No?
Maybe
Is that a
bryozoans.
sponge?
Or maybe, I'm not sure. Maybe bryozoans. I mean, there's a sponge, of course. This is the at the right side. But this organism that looks like algae or fungi is probably a bryozoan.
No, no worries. You've got a lot to juggle.
Yeah, I'm down.
We knew the downward slope, we wouldn't get much view, so just just worry about the vehicle, don't by us.
That's good, thank you.
Person in the chat was pointing out that the sponge looked like the Phareide, and indeed it looks like this. Could be something similar.
That's fine. They're both familiar.
Both familiar?
Yeah.
Which things are Yeah.
Such a steep drop off.
Yeah. No, don't worry about it right now. When we set up, yes, they'd be on the front. So
just coming down the other side of that ridge. This is obviously approaching the bottom where all that falling sediment is collected. Sediment's really cohesive. It's really gripping to the rocks, even on these quite steep faces. It's very fine. It sort of sticks to itself. Quite cakey. And now we've reached the bottom where it's all collecting. And the surface looks quite different here. This looks like a lot of material on the surface.
Our shift is coming to an end, so I say thank you to all people listening to the chat, and also thank you very much to the pilots. They did an excellent work. It's very difficult to navigate here with these rocks. Now Sophia will take over. Thank you, bye.
Incredibly complicated terrain. Even though this is covered in sediment, this is undulating with some quite steep drop-offs. A reminder of what our team has to contend with. Oh, something in the water here. Is that octopusy? Could we have a zoom on that? Oh, it's one of those, are they seabed?
Yeah, it does look like a novella, yeah.
Novella.
You're right.
Oh, I thought that was a squid or an octopus in the water.
Yeah, me too.
Nubella's pretty cool too.
For a second, yeah.
That's great, thanks, Kay. It's a reminder of what the marine crew have to contend with here. A vehicle the size of a bus, tethered by 1,000 meters of water to a vehicle on the surface. We have to dodge this tricky terrain while making awareness of our umbilical, not wanting to get tangled or caught in anything. And then there's a vessel on the surface dodging icebergs and sea ice, which move in different directions because they're moved around by different forces. So a hugely technical operation, but pulling it off. And even beyond that, actually, the hugely technical operation of, there are people at home watching us explore new seabed in Antarctica. And when I started my career, that would seem like magic. That would seem impossible. I can remember sending one email a week.
Yeah, go ahead, ROV.
Hey Pete, would you be able to bump up to 0.6 for the speed?
Yeah, no worries at all. Going up to 0.6 now.
Thanks, sir.
Oh, there's something interesting here.
Oh, nice. It's coral.
A lot of coral. Can you swing around and take a look? this is nice. So obviously we've got a face here, a rock face that is nicely aligned with the current and is just prime real estate, because we have not seen corals this big in a while. Okay, I'm just going to have a shift change so I can go grab some dinner. So thanks for joining us, but stick with us. We're just changing shift. Are you still there? Are you still the smaller team?
Sorry.
Are you still the smaller team? Do you want to come back after dinner?
No, it's okay.
Sure.
We're just gonna have an hour.
Oh yeah, we're coming up then.
Is anything particularly full that I should not touch?
Just that there's a scallop in that one, which unfortunately kind of means we don't
may
have
try.
those. So if you find something really lovely, you can try for
We're
it.
gonna try. It's actually supposed to have pretty good food.
Yeah, but there's things like brachiopods, so it's not going to be a problem.
Okay.
Took a
Oh,
few
cool.
more
We can put
rocks.
stuff on coral quivers as well.
Oh, that's true. I've not really made use of those.
Okay.
And just that these two are reserved now for the way back up.
Okay.
15:30.
Great. Thank you. Have a good dinner.
Thank you.
Oh, that's beautiful. Let me just double check if I don't have any requests. Nope, seem to be okay. Can we just go a little bit more around it and then we can continue to the point. This is beautiful. Wow. Can we have the laser sign? Yeah, sorry. Just for yeah, some good shots. Just for in case we want to measure it later and quantify it. It's gonna be super useful. Oh, this place is beautiful. Yeah. Very different from yesterday, huh? It's very interesting. Okay. Yeah, we're good. Yeah, we can continue on the way to the next waypoint. Are we still down the slope, I guess, a little bit?
Well, it's
Kind of bumpy, right?
all over the place.
Okay.
So
We'll figure out.
follow a ridge and drop down,
Mhm.
came up and then drop down.
Okay.
Bridge, are we?
Yeah, go ahead.
Updated with my date.
Excellent, thank you.
And after Tom and Pedro, it's Sophie and Andres here. We continue our exploration of this beautiful site of today. We started in the morning with beautiful walls of sponges, and we are going towards the last hour of the dive with these incredible walls of corals and more sponges. We have been kind of crossing this mound of the seafloor, this large rocky structure we've seen most of the day. And as we cross to a new kind of upwards location, we might have a bit of more flat areas, but we are expecting to have an overview of another wall just before the end of the dive. To see if, for example, the orientation of the sites we're exploring do harbor or not different kind of communities along the way. This area is clearly quite rich in organic matter, so the food for the organisms we see on the seafloor, and the currents seem to be quite strong, and particularly in some locations. So I think that's why sometimes we have certain communities in one of the sites, at least, of the locations explored. Earlier today, just before my break, I was looking at a beautiful wall full of corals, and it was several meters high, and sponges, and this place was just clearly in the middle of the channel we're exploring, and I think this kind of conduits bring food to these organisms that they hang in there and grow up preferably on those locations. The glass sponges here are just incredible. Look at the size of that thing. Wow. It's beautiful. Oh, and we have another black coral there. Can we just have a quick zoom in? I think it's the same we saw earlier today, but just want to double check. Yeah. Yeah, then we cannot really clear. No, it's okay, we can continue. It did seem quite similar to the one we saw earlier. Go ahead. Yeah, definitely yes. It's gonna be zigzagging out of the waypoints, I think. it, yeah. That's perfect. Nice. Yeah, looks like the greenery, the kind of greenery we saw yesterday. Look at that. It's incredible. Wow. This place is amazing.
All this is like a cross bridge, are we?
We can.
We
Yeah.
move pause, please.
You're going downwards, right? Yeah, we can come back up slow.
Alright, big rig, sorry, it's at the front, but yeah, we're slowing down to a stop.
Thank you, Jason.
Because otherwise you're gonna go downwards, right? So. No worries, it's okay. You can come back up slow. Yeah, we can continue to the next waypoint. Because it's gonna be going along the ridge anyway. Let's see if we can. Yeah, that's what I was thinking. Just a little bit, yeah, maybe a little bit like that, and then if we can try to have a camera at least halfway to the bridge.
Why would come down on the face?
Yeah. Not a problem for navigation. Okay, then that's perfect. Even just that camera has an incredible view from down. Oh. A lot of. Most of the corals seem to be the ones we've been seeing today. Just seeing. Wow, look at that massive spot.
Copy that.
Yeah, I think we can orient, go back up, yeah. This gives us a good overview of the site. Did you see any squids hitting the RV today?
Not yet.
You did? Not yet. We were talking about trying to access that camera feed the other day, so Tom could search for it.
Yeah. Has he found anything yet?
I don't know. We did keep the timestamp so we can see if it matches for what it was seeing in this camera during the night. So. Oh, nice.
No squid, just this octopus.
Well. I hear during my break that there were seals around the ship. And it was a beautiful video of just swimming along the ship. Like an hour and so ago. Yeah. Wow. They're always cute. themselves. Yeah. We've seen quite a few, not just scallops. Sometimes just leftovers of other sea urchins even.
Well, that's.
Yeah. I would expect this side, this part of the transect to be more flat. Let's see.
Yeah, this is pretty exciting. Thanks.
Yeah, it is. Come on. Look, there's a rock full of sponges ahead. We were looking for those sea pigs in the morning. Now they show up. See? It's always like that. And the next waypoints are upslope, I think. If we see some. We just want to have a peek and see before we come up at least what's on that side.
Okay.
Oh, what is that? Sorry. Ah, it's a greenoid. Well hidden. Perfect camouflage. No? Thank you. Yeah. There's, yeah. You can have a look. Mm-hmm. The sponge ridges, huh? Oh. Let's see if we can. We tried that yesterday, not today. Oh, nice. Look at that one. Look at this one. Has something inside?
Go ahead.
It's not big waypoint, Jason.
Copy that. Are you happy with the speed at the moment, or do you want me to slow down?
Oh, interesting.
Slow down a touch.
No, that's
Yeah,
okay.
you go to 0.4, that'd be
Sorry.
great, thank you.
That's good.
Beautiful. Mm-hmm. is beautiful.
So I'm gonna have to turn right.
Mm-hmm. Oh, nice. Can we have a quick look as well after to the corals just here on the left?
Sure.
After the jellyfish, because the jellyfish is great.
Pause the ship.
That's awesome.
Bring it to Johnny.
Go ahead, RV.
We can pause on that, please, Jason.
Yeah, can do.
Listen to piano music up there.
Yeah, like it's kind of serene.
Alright, very soothing.
So I'd like to have a quick look at those here. So I think we're looking mostly for the light yellows. So let's see if we see them on either side. Wow. So this one?
Probably.
This one might be right in front of the little one.
This one? Tell me which one. I was looking at this one, which had a lighter coloration from afar.
Yeah, I think rotate over to the right.
Can we just move slightly to the
Move
right side?
sideways to the right. I think it's the right of the starcher colony, if there's one. Yeah, these are kind of darker, but I
Mhm.
think
They look
I think it's
similar, yeah.
Yeah, like this one here. So yeah, I think these might all be these two.
Okay. This one different? Somehow?
I think they're probably color variations of the
Yeah,
same species.
because they do
but-
seem to be from the same kind of large colony, but I'm not sure. We
Yeah.
can take that branch out. This one.
Sample that.
Yeah, we're gonna sample that for a coral quiver.
It's just we started all of a sudden seeing the color variations, and it could be color variations, or they
Yeah.
could be different species.
Look
but
here, it looks kind of a variation within the same
And then
colony.
it has the dark orange,
Yeah.
but the light ones seem to always be light.
Okay.
So yeah.
We'll pick that one.
Thank you. And if you see an anthomastus, we passed a red
Yeah.
one that was all
Yeah.
closed. So
It was.
just
We
keep
already had passed when I
Yeah,
sent a
that's
message.
fine.
That's
Yeah,
good.
that's really fine. No point going back for it.
But
if
you
see
would
another
want,
one.
you want the whole colony?
Yeah, with anthomastus, you kind of have to take
Yeah.
the whole thing. So
Yeah. Okay.
thank you so much.
There's one there. I knew it. So we're aiming for the one of the coral quivers that's still empty. I think you have to be the ones on your right side. And I'm not sure if 5 or 7 is still occupied.
Yeah, I think 5 and 7 are still open.
They're still open? Yeah. Okay. Then we can use either 5 or 7
Yeah.
or the right side ones.
So you want the whole floor?
No,
No?
we're just gonna cut the labels.
Perfect.
So first we want to zoom in, have a good look at the colony. So we're looking for this branch here. This one. Oh, it has a, I think it's a nidroid maybe. Oh, that's beautiful. And if we can also towards the bottom to see if where kind of the benching happens. Thank you. Yeah. And just before we start sampling, I would like, if we still can, there's one just here that might be still inside as well. Yeah, just have a good look at the smaller ones. Thank you. And then yeah, we can collect that one, that little piece. Yeah, that one on top. This would be for coral quiver 5 or 7. Which one is easier?
So you think of this one,
That piece. Yeah, that one. Thank you. Can we have a zoom in as well while it's still in the arm? Nice. That hydroid is beautiful. This is awesome. Yeah. Thank you. I can put it back. Yeah, I think so. Extra zoom. It's beautiful.
Look at it, so?
Is that okay?
Sure, you got it.
Are you locked, Tyler? Or are you just holding it?
Frozen.
Is it? Weird one. Ah.
There you go.
That's incredible.
Cool.
Yeah, thank you.
There's wants a couple from here to do some food stuff, so can we grab send a dark orange
Let
copy?
me just do a few zoom-ins before we continue.
Thanks. That way we get different color morphs, and if they are genetically different, we'll find out.
Yeah. So actually, after this one, no, we won't mix it. It's okay. We can cover this one, but we can open the other coral quiver because we want to take a few pieces from another colony as well to complement. So
Can't mix them.
well, yeah, we will avoid to mix them at this point.
Put this up on the next one.
We're just gonna collect a few more pieces of the colony around, and then after that, also do a couple of zooms just into the animals in there, and then we charge. Yeah. I see something super interesting. Then zoom in. You see this guys over there? Just so we can zoom in there. That's one of the places after.
Hold on one second. Yeah, I'm just getting that 4 down. Start 3, so.
Yeah.
Okay. Alright, stop there. Stop.
Just want to have a look at here. Oh, nice. Those are beautiful isopods. Just gonna have a quick note and take oh, that's awesome. Those are alien-like. We have an ID from an expert from earlier on. This was a recently described species. That was really cool. Thank you. Thank you. So I'd like to just to do a couple of zooms on the rock before we collect the rest of the coral.
Sure.
Just want to see here, like around the rock here and here. As we get the tiny stuff, let's say. Nice. Okay. It's just a price on. Oh, a beautiful snail. Okay. Thank you. Just a bit upper, though I think there was also some little amphipods, but maybe I'm mistaken. Yeah, there's a few of them there. They're the same, I think. Nice, thank you. Okay, so now we're gonna take 2 pieces of this orangey yellow coral. Ideally, one from a different colony, so we can pick up, for example, from here and from here. On that branch, yeah. So just a length of about 10 centimeters, and then we can put them together on the same focus. So laser size.
Of this one.
Yeah,
Hold
okay.
on. You want that one?
Yeah, we can do this one, and then from here, anywhere that is
Okay.
easy for you. So that one we're probably gonna have to go.
Just take it to the bottom?
Yeah, I think so. Based on the size, yeah.
Yeah, it's a shoulder down.
Gotcha. Cool. That is a good shot.
Thank you.
Let me show this.
Yes. Mm-hmm. Always have a close-up before we put it back on the coral quiver.
Frozen. Cool.
Oh, it has an amphipod as well. Thank you. Is it quite? Does it fit, you think? That's good use. Mhm. Mhm. There it goes. Okay, thank you. And now can be from the other colony that was just there on the side. Just a fragment from this one. If it's easier from this one, it can also be. So if you're wondering why we're taking several fragments of the colonies just here, is because one of our colleagues, for example, wants to have a deeper look into the feeding preferences of this coral, so she will be using different specimens for confirming her observations what do they feed on? So we call them the food web studies. So if you hear us sometimes saying, "Oh, that's for food web," that is because there's that's focused besides just besides describing the new species and besides confirming distribution of the species we already know. So this is an additional as well point we try to explore. Fine. Yeah. Just so before we also move too much and start sediment all around.
Yeah, there's definitely a place already quite nice.
Just go ahead. All you. So Alex has some requests.
There are just 3 different samples taken, and 2 put in the same coral pit.
Yeah. I thought it was for food.
But 3 samples and 2 coral pitters.
No. One is separate. The first one and then the 2 others are
Yeah, so there's 2 samples and 1 coral pit. So never ever. I know she says it's okay, but I can't do the genetics with it because they're the same. And then they touch each other and they slime on each other, so she gives it either. So,
Yep.
yeah.
I'll make sure.
Yeah, so it's the orange colony was the other one we needed. So.
No, we're gonna take a look at this. So now it's doing this thing. If possible, I might need to be. one? Okay, that one.
The smaller plants too. No,
That's the big. See?
There's another one. That looks like.
Yeah, we can try.
Because I need those
Yeah.
crocodiles. That would be the ideal size.
Yeah.
Just
Thank you.
something like this.
Yeah, yesterday was a monster.
That is back into
Yes. Was.
So, ascend for a minute. So I guess we have like 10.
Did you say it?
Yeah, as much time as you
More
want.
and less. No, we should be on like at 8 because it has right after another dive. So we're gonna make an effort to make sure we are on time. We collect the rock and then after Alex finishes his shot, and then we come up, I guess.
Oh, yeah.
But it doesn't make the same awesome noise. Like grinding pepper over there.
Those are so cool.
Better.
That's incredible. And yeah, literally they're just feeding. Look at that. Yeah, it's okay.
soul cheese that.
Honestly, maybe after this, 10 seconds.
Look at that guy. No, lindo. Look at the colors.
Yeah.
It's beautiful.
I think it's adorable.
They're having fun.
That's the most effective.
That is incredible. Good.
Okay.
Some rock sampling time before we come out.
After the, oh yeah, probably. Because of the text on friends, sometimes they take a couple hours.
Small piece. That.
preference?
I don't know, maybe too big.
They're too big?
What about the one on the left?
I'll try this one.
You wanted that one?
This.
Go for that one? That one.
Yeah. Let's go try that one.
No, the other one came with this one.
Just give it a look. Yeah. See if it works. Too far. No.
Here?
We're just gonna see if it's.
Sí,
nice.
sí, sí.
Yeah, okay. Right.
Pink is attached?
No.
They're gonna have to accept here.
So you're gonna show
Let's
that.
accept the big one, I guess.
Yeah.
Yeah, it's too. It's okay. We can. We'll put the other one. No worries.
It's loose.
It's loose, yeah.
Well, if the other is okay.
Yeah.
If it's too difficult, the one on the right is okay.
So just kind of shove it out. Pick it up off the seafloor.
Hmm.
The one on the right is okay after the sponge.
He's gonna try another technique. Just push it on the side. There you go. Nice.
Is that okay?
Feed it right away.
Good? Okay. Right, we can wait.
Ah, yeah.
Let's turn it.
Is that what we're going?
Oh, now. It's on the side.
Yeah, there.
Just kidding.
All that
Want me to
work?
move over?
I'm good on the porch, right? It's close. Yeah, we just kind of gotta bring it up.
They're definitely watching seals, I'm sure. Yeah, that's not it. can put it on the rock basket, if it's possible. The one inside,
Inside, alright.
on the porch.
Clear?
Should be clear. I'll take out the sponges. All good?
We can also go right off bottom, no problem. Yeah, not a problem. We don't have any more sampling of animals here, so it's fine. No, not at all.
Alright. Rock. Secured.
Great. And we are good. We can come up. We're gonna have 2 stops on ascending at 50 meters and 30 for
Thomas one.
water sampling, and that's it. So we can come up.
That is the unlock. Hey, happy birthday, buddy.
Happy birthday, buddy.
Happy birthday, Carlos.
Thank you.
Parabéns.
Sit here.
Portuguese is better. Parabéns in this case.
You have to tell them cute.
Oh, that's beautiful. Bye, beautiful seafloor.
Really,
In 15
Darby.
minutes.
Go ahead.
We are off bottom, Jason.
ROV up bottom. Copy that.
Yeah.
Hey, good.
Yeah, did you want to do a sign up?
Yeah, okay. We can do it together.
Cheeks.
That's what you get when you have 2 Sophias.
Yep.
Okay, so we finished our sampling at the moment, and you have to do with 2 Sophias at the same time.
Yes, we do.
First, I'd like to say parabéns, Carlos, in Portuguese, because, well, we share the same language. We're gonna be off bottom. Well, we're gonna be on deck in about an hour, maybe.
Mhm.
So until then, we have a bunch of scientists getting prepped with all their containers and labels and so on. Tomorrow we are planning another time. Actually, not from tomorrow, in 3 hours or so.
Yes.
So
We are doing a very quick turnaround and heading straight back in. So keep those YouTube notifications on because you'll be the first ones to know when we're back live.
Thank you
Yeah.
so much. See you soon.
Thank you. Enjoy the ascent.